Okay, good afternoon, everyone. I'm Marco Bottino. Can I be heard in the back? Okay. So I'm Marco Bottino from University of Michigan School of Dentistry. It's a great pleasure to be here with all of you today. And I would like to thank my colleague, Professor Shahwa Liu from University of Texas A&M in Dallas for helping me putting together this symposium. So I'll be one of the speakers. I'll be speaking last. Together with me and Professor Shahwa Liu, are Professors Juanita Ono, she's at the Houston School of Dentistry, and Professor Zoe Zhu from Tufts University. So the logistics of the symposium, so each speaker will have no more than 20 minutes, and we'll have within the transition time for one quick question, and then we'll have in the end, a panel discussion for another 10 minutes or so. Thank you very much. So I would like to introduce Professor Juanita Ono. Thank you very much. Thank you.
Hello, everyone. Can you hear me? Yeah. Nice to meet you, and thank you for opportunity to present my recent work here. I'm from University of Texas School of Dentistry at Houston. And today of my talk, we're going to talk about the alveolar bone loss and rescue under the modulation of PTHRP and Hedgehog signaling by the dental follicle. Okay, so I'm not periodontist, but I'm an orthodontist by training. So here is a typical case that I see every day. And he comes to me, he's a young boy who wants braces. He has a little bit of crowding, spacing, impact canine, but yeah, it's relatively straightforward that we see every day. Within 18 months, we can have a reasonable occlusion for him. However, not every case are like this. I understand that because of his healthy periodontium underlying the tooth movement, that allow me to have the good outcome like this. But what about in the case like this, where you have a loss of periodontal ligament or periodontal bone surrounding the tooth? We cannot move these kind of teeth. Or even the case like, I try to put the laser pointer, like this, where you have an implant there and patient is deemed to have retreatment of the orthodontic treatment. We cannot move the implant. So we cannot do anything. Or the other rare case, such as some genetic disease that doesn't respond to orthodontic treatment, such as PFE or primary failure of tooth eruption, which you do not have the bone surrounding the root, so you cannot move the teeth. Or other disease or of the patient that receive medication that do not allow tooth movement. So what should we do in this kind of case? So understanding how periodontal development occur will be the key to understanding how, for the regenerative therapy in the future. So we focus primarily on the dental follicle, which is particularly the dental follicle cell that express parathyroid hormone related protein or PTHRP. Dental follicle cell is, dental follicle is a sac-like membranous tissue that surround the developing tooth bud. And it has been previously reported that PTHRP is expressed by the dental follicle and required for tooth eruption. And we previously reported that these PTHRP expressing dental follicle cell differentiate into the periodontal ligament cells, cementoblasts on the acellular cementum, and also osteoblasts of the cryptal bone area. And this formation is controlled by the signaling pathway by the PTHRP and its receptor, which is the parathyroid hormone receptor 1, or I will call it PPR. So therefore, I would like to show the expression of the PTHRP, and which is the ligand, and also the PPR, which is the receptor within the developing molar. You can see in the figure 1. This is the developing molar of the mice at the postnatal day 3, where the tooth root structure start to form, and also include the periodontal tissue. So this is the red indicate the endogenous expression of the PTHRP, and this is a PTHRP emissary knock-in mice. So you can see that this red fluorescent expression is limited to the dental follicle surrounding the developing tooth bud. And if you expand this area where the root will form, you can see that these cells are in the different domain than the herds or the epithelial cells. And what about the receptor? This is the immunostaining of the molars of the mice at the postnatal day 3. And you can see that the receptor is ubiquitously expressed in many cells within the developing tooth bud, including some of the pulp cells, odontoblasts, and alveolar bone osteoblasts. But more importantly, it is also expressed in the dental follicle in the overlapping manner with the lichen, the PTHRP. So what about hedgehog signaling pathway in the tooth root development? Hedgehog has a lichen. I mean, the lichen that is expressed in the tooth root development is the sonic hedgehog. And sonic hedgehog mainly expressed at the herds, which is the signaling center during the tooth root formation. So when the crown is formed, the outer and inner epithelial will fuse and form the herds right here. And these herds structure express hedgehog or sonic hedgehog. And sonic hedgehog usually binds to patch 1 or patch 2 receptor. And previous report show that the patch mutation mice have a disturbance in the tooth eruption and tooth root formation. And also Shai's group also show that both inhibition and constitutive activation of the hedgehog signaling in the GLI1 root progenitor cell leads to shorter roots. However, what about the hedgehog signaling pathway and their effect on the dental follicle? How these signaling pathway induce the periodontal development from the dental follicle is unknown. So here is a simple diagram to show the hedgehog signaling pathway in the condition that hedgehog doesn't bind to the patch 1. The patch 1 has inhibitory effect on the smoothen G-protein coupling receptor, and which allow the hedgehog pathway to occur. But when the hedgehog bind to patch 1, this inhibitory effect will be suppressed. So therefore, the smoothen signaling pathway can occur. And transcription factors that induced by the smoothen pathway is including GLI1, GLI2, and GLI3. Okay, as our research strategy, we utilize 3 lines of the mice. The first one is the PTHRP CREAR lines, which we can specific label the PTHRP dental follicle cells. And the reporter mice, Rosa 26 tomato reporter mice. And the third one is the patch fox mice. So for the experimental model, we give tamoxifen to these compound mice at day P3 when the root and periodontal tissue start to form. And then we analyze micro-CT, histological sections, and the bulk RNA-seq during the periodontal development. So what happened when we give tamoxifen to these mice? So the Cre recombinase will translocate into the nucleus and remove stop sequence at the Rosa 26 tomato allele. And simultaneously, it will remove the critical exon within the patch 1 locus, which is exon 8 and 9. And as a result, the PTHRP expressing dental follicle at day P3 will be red, expressing the tomato fluorescence. and simultaneously lack of patch 1. So by doing this, when they multiply and differentiate, you can trace the cells over the time of the periodontal development. And remember, patch 1 deletion caused the constitutively activation of the hedgehog signaling pathway. Okay, remember, because patch is inhibited, it has inhibitory effect on the smoothen. Okay, so now let's move on to the result. So on the left-hand side, this is the molars of the mice, 3 months old. And you can see that the PTHRP cells at day P3 can develop into the periodontal ligament. And also there's osteoblast on the cryptobone that also express red fluorescence. But when we delete patch 1 or overactivated hedgehog signaling, you see a clump of red cells between the interseptal area, and also there's less alveolar bone osteoblasts at the interradicular area. What about the micro-CT analysis? And you can see that in the overactivated hedgehog model, there's a bone loss at the alveolar bone area, as indicated by the height between the CEJ to the alveolar bone crest is increased both in the molar M1 and M2. So therefore, this result suggests that the constitutive activation of the hedgehog signaling in the PTHRP expressing dental follicle induced alveolar bone loss. Okay, what about the periodontal ligament side of development? So you can see here, we immunostain with the perostin, which is a pretty good marker for the periodontal ligament. And you can see that in the control, which is the head group, you see the periodontal ligament that co-express the perostin, and they are descendants of the PTHRP dental follicle cells. But in the knockout, the patch 1 knockout, you rarely see the cells that develop into the perostin positive periodontal ligament cells. So let's take a closer look at the area. So this is the interseptal area. So you see alveolar bone at the septal area with the red osteoblast. And also you see the periodontal ligament that co-express with the perostin in this area. And at the interradicular area, you also see the osteoblast in the cryptobone in the control group. But here in the patch knockout group, you see these clump of cells and very few amount of cells that express the perostin in this area, and almost no osteoblast, red osteoblast at the interradicular area. So these results suggest that the hedgehog activated dental follicle cell fail to properly differentiate into periodontal ligaments and also the bone at the crypto area. Okay, so the next question is that what would happen if we inhibit these overactivation or suppress the overactivation of hedgehog signaling pathway? So we use a smoothen antagonist, LDE225, which is a common drug for treating basal cell carcinoma. And these act directly on the smoothen pathway, and it suppresses this hedgehog signaling pathway. And we give this drug into mice during the periodontal development. And you can see on the right-hand side, we can rescue the development of the PTHRP expressing dental follicle into the periodontal ligament that express perostin. And there's a increase of the bone area, alveolar bone at the interradicular side. Okay, lastly, we also did the bulk RNA-seq. So what we did, we harvest the dental follicle from the P8, and we enzymatic digestion to get single cell suspension, and we sort only the red cell, the red RP dental follicle cells, and we did the RNA-seq, the ball RNA-seq. And in the right-hand side, you can see this heat map showing the potential target gene under this signaling pathway. The red represent the increase and the blue represent the decrease. And you see that the hedgehog signaling pathway involve the genes that involve in the hedgehog signaling pathway, like such as GLI1, PATCH1, HIP increase. Also, dental follicle markers such as ecto2, spondin 1 increase. And on the other hand, the markers involved in the periodontal ligament development, such as scur axis or periostin, decrease, and also the osteoblast differentiation markers also decrease. Interestingly, the FOX family, which is a previously reported transcription factor that involved in the hedgehog signaling pathway in the neural crest cells, also increase in this model. So in summary, I think I convinced you that the constitutive activation of hedgehog signaling pathway in the PTHRP expressing dental follicle cells impair the differentiation of these cells into the periodontal ligament and also osteoblast, and that led to the alveolar bone loss. And the suppression of this hedgehog signaling pathway is required for proper cell fate development of this RPDF cell. And FOX transcription factor may act as a downstream of this hedgehog signaling pathway and regulate the differentiation of the RPDF cell. So in conclusion, the inhibition of hedgehog signaling pathway is essential for the RPDF cell to properly differentiate into the PDL cells and the alveolar bone osteoblast. And that leads to the formation of the functional periodontal tissue. So there's still some remaining questions and future directions that we are currently studying, which is what is the consequence of the hedgehog-induced compromised periodontal attachment alveolar bone? So what would happen in these mice if we create the periodontal injury? How would it heal? Would it heal similarly or different? So that's something that we are pursuing right now. And also, what is the role of the FOXF1 transcription factor in the PTHRP hedgehog regulation in the periodontium? And also, can hedgehog inhibitor can be applied to promote the periodontal attachment formation, because the LD225 is an approved drug, commercially available. Okay, so to finish my talk, I would like to thank the lab member of my group, especially Misuki Nagata, who's a very talented postdoc who drives this project, and also Seth9, who's previously orthodontic resident and who helped with the 3D analysis of this project, and also funding from the NIDCR and the AAOF. Thank you.
So as we switch presentations, we can, Dr. Wano can take one question from the audience.
We do not see phenotypes at the herds, because, and that makes sense because, well, in a sense, because we didn't in the follicle yet, but that's a really good question. What would happen if some hedgehog is removed in the digital sense? It gets the same result. So that's a good thing.
Thank you. Thank you. So our next speaker is my co-chair, Professor Xiaohua Liu. Thank you.
Thank you for the introduction, and I'm very glad to have the opportunity to present our work here. The research in my life mainly focus on two aspects. So first, we are very interested in design and develop the bio-inspired materials for dental tissue regeneration, including pulp and periodontal regeneration. And we are also very interested in developing new technology and 3D platforms to study cell material interactions and try to use the biophysical and biochemical cues to control stem cells fate, like the periodontal and ligament stem cell and dental pump stem cells, the polarization and differentiation. But today, many focus on the first part. I'll tell two stories. One story is about the design the bio-inspired materials for real bone regeneration. And the second one talk about the periodontal ligament regeneration, especially we focus on the sharpest fiber and the PDL principal fibers regeneration. So in the first one, I talk, we develop immunomodulator ECM-like microsphere for alveolar bone regeneration in diabetic condition. We know that diabetes is one of the most common metabolic disease. It affects more than 30 million people in US alone, and more than 60% of the diabetic patients also have periodontitis, which means these patients also have the alveolar bone loss. And currently, there are very limited studies on alveolar bone regeneration on the diabetic condition. And among these very few studies, most of the work focus on the osteoblast cell or osteoclast cell. But we know that diabetes is a chronic inflammation disease. So we think that instead of targeting on the bone cells, target on the macrophage or the immune system, maybe it's a more promising strategy for alveolar bone regeneration. So in this study, we tried to develop a hyper-modified gelatin-based injectable microspheres for the alveolar bone regeneration. And you can see this microspheres is self-assembled from the gelatin, which is from collagen, and it's composed of the fibrous structure that can provide the physical cues to promote osteoblast cells proliferation and differentiation. And we also used the heparin here to interact with the gelatin so that this heparin provides a binding domain to interleukin-4. So in that way, it can protect the degradation of interleukin-4 and also control the release. And use this cytokine interleukin-4 to change the fate of macrophage from M1 to M2. So in that way, it may work to change the microenvironment in the defect area. So that's all experiment design. And this is the materials that we synthesized. And this is microspheres we want to use. And it's entirely composed of the nanofibers, use gelatin. And because of this structure, so this material will have very low density. That means when the material degrades, it has a very small amount of the byproduct. And it has a very high porosity that will provide the more space for cell attachment and also cells growth. And we use these spheres as a control group for study. And this slide shows that these spheres have very good biocompatibility. Cell can easily attach and grow on the surface of the of the spheres, and you can see very interestingly the cells, once it attach on the surface, they can grab the microspheres together. So in that way, when we use this like microspheres as an injectable cell carrier, we do not need either a cross-linker in vivo. So that can, compared to hydrogel system, that is a benefit for us. So we can see from the image that the interleukin-4, this cytokine will be very evenly trapped into the microspheres, and because we added the heparin to modify this system, so these microspheres can very well control the release of interleukin-4 from the microspheres. You can see from the release profile, after we modify these spheres with heparin, the sustained release can last for over 3 weeks. And so the next is testing the bioactivity of the interleukin-4 released from the microspheres. And we first treat the macrophage, use the LPS and IFN gamma. And after treatment, you see this marker, CD68 is a macrophage time marker, and CD206 is for M2 macrophage. After treatment, there's no M2 macrophage. But if we add the interleukin-4 that is released from the microspheres at day 1, then we can see a significant number of the M2 macrophage. And similarly, if we treat the sample with the interleukin-4 that is released at day 14, it also can show large amount of the M2 macrophage. So this experiment shows that the interleukin-4 released from the microspheres can retain high bioactivity. So after this in vitro study, the next step what we do is we generate a real bone defect model using a diabetic right model, right. And then we implant our microsphere system into this defect and see how the bone will be regenerated in this defect area. So this is the 7 days result. We first observe the distribution of M1 macrophage. And you can see this is the experiment group, and this is a diabetic group. So if without the treatment, we will see a large number of M1 macrophage, but after the treatment, we will see significantly decreased the number of M1 macrophage. And similarly, we see the increase of M2 macrophage in the defect area after the treatment. And this the quantitative data shows that the ratio M2 to M1 significantly increased after we treated with this microsphere system. And you see this ratio is very similar to the normal rights. Okay, so these are the 2 weeks results, and we examined the TNF alpha, this factor, and you see after this treatment, this TNF alpha's expressions significantly decreased compared to the controls group. And this is the quantitative data. Okay, and another one we testing the osteogenic differentiation marker. We examine osteorex expression. You can see that after 2 weeks, this expression is much higher than the untreated groups. So that's 2 weeks. And another marker is for the ALP. ALP, we get similar results that the ALP is expression in the interleukin-4 loaded microsphere system is significantly higher than the controls group. So the the last one we treated it for the 4 weeks, and we examined the defects of bone formation, and this is a macro CT, and this is a histology. You can see of the treatment, most of the defect area has been regenerated with new bones, although we still can see a little bit of this like that is the microspheres that are not completely degraded yet. So overall, we see that this system did work by switching the fate of macrophage. We can change the macro environment and help the bone regeneration. And I will give a brief summary for the first story I just talked. So in this work, we designed and synthesized a ECM-like microspheres and use this microsphere as an injectable immunomodulatory biomaterials. And this interleukin-4 loaded microspheres can effectively switch M1 macrophage into the M2 phenotype, resolve inflammation, enhance the osteogenic differentiation and the new bone formation. So this work shows that the development of immunomodulatory material is a promising approach for bone healing in diabetes. And then this work was, right now our continued work is to add, the next step is to add the periodontitis to see if this system is still working. Okay, so the next story I'm talking about is about the Sharp's fiber and the periodontal ligament regeneration. We know Sharp's fiber are a portion of PDL collagen fibers that are embedded in the cementa and the bone. And right now, in my point of view, that the major challenge in periodontal regeneration is the interface. That is, means the regenerative tissues between the PDL cementa or the PDL alveolar bone. And Sharp's fibers are the structure that connect these interfaces. So when we first started the work a few years ago, we look at the literature. We try to find the characterization of this Sharp's fiber, but very surprisingly, we found there's very little information about the anatomic parameters about the Sharp's fiber, such as for the Sharp's fiber, what's the diameters, what's the density, or the length, or the orientation. This kind of quantitative information are largely missing. So in our work, the first step, we are trying to do a comprehensive and quantitative analysis of the Sharp's fiber. We use the right model to do that work. And next, once we get that parameter, we're trying to develop the biomimetic matrix and to induce the Sharp's fiber and also the PDL's regeneration. So we developed two tools to calculate the Sharp's fiber. We use SEM to calculate the diameter and density of the Sharp's fiber, and use the light microscope and combined with the Cyrus rate staining to calculate the length and the insertion angle and embedded angle of the Sharp's fiber. And this is available bone side on the Sharp's fiber. You can see the Sharp's fiber bounder as we indicated the layers here. And we also found that like from the cemento side, the Sharp's fiber, this like density length is very different. And we did a very comprehensive calculation of this Sharp's fiber, including different teeth on the same teeth, different like area, and also for the different groups. Here, I just give a small portion of our calculation. And overall, we can see the Sharp's fibers bundle, like between the bone side and cement side, the range is about like 5 to 6 micrometers. And bone side, the number is a bit larger than the cement side. And for the density, the bone size density is significantly lower than the cement side. but the length of the sharpest fiber on the bone side is about like 3 times larger than the cement side. And the insertion angle is roughly about 50 degrees. All this number is from the mandibular first molar, and the fibrous group is the oblique group. But the other group, they will have different numbers. But we use these parameters. Next step is to design a scaffold and try to mimic these parameters. And here's the biomimetic matrix that we design and synthesize. So we're starting from the elect spinning of the gelatin and form the matrix. And the next step, we use the laser ablation approach, try to generate this tubular structure. And our hypothesis is we will use this like tubular-like structure to guide the PDL stem cell to growth, and then inside of this matrix, they'll form the PDL principal fiber. And on the interface, this area, they will form the sharpest fiber. And here's the results you see is our synthetic matrix. That this one, each like tubule, each hole is represented like the. Later on, we expect it can form a sharp fiber bundles there. Okay, and we can mimic the density of the sharpest fiber. Then also the diameter of the sharpest fiber. And from this confocal, we also try to mimic the insert angle of the sharpest fiber. You see here, it's not like 90 degree perpendicular. We can generate this 3-dimensional matrix with some insert angles. So next step, we'll see the PDL stem cell on the surface and see how the cells will migrate and form the PDL principal fibers. And this is the result about the PDL cells migration. You can see from the top view, the cells are very well aligned compared to the control group. The control group means that we still have the fiber structure, but we do not have the tuberous architecture. So because we have no tubular structure, so the cells only stay on the surface. But with this like tubular structure, you can see cells not only stay on the surface, they also migrate inside the matrix and even to the other side of the matrix. And this is showing the dynamic process. You see the cell, when we see it, it is supposed to stay on the surface and then slowly migrate inside the matrix. And then after 24 hours, it can evenly distribute inside the entire matrix. So this is the result of the PDL markers. We culture this like a cell matrix construct for a total of 14 days, and we staining the collagen type 1, 3, and the peroxidine, and we can see as time goes on, this max expression like increased, significantly become more and more stronger. And we also found the collagen fiber bundles formation inside this into this tuberous matrix, and we use polarized microscope to see this collagen fibers formation. We also found a very interesting result is that this tuberous structure actually prevent the osteogenesis of the PDL stem cell. You see these 3 markers, RUNX2, OCN, and SP7. The expression significantly decreased compared to the control group. That means there has no tuberous architecture. So the last study we did is we we had a preliminary like experiment to do the in vivo testing. So we implant this matrix combined with cells and in vivo, and this is the 4 weeks results. This is just the preliminary data. We can see after that 4 weeks, we see some very good like PDL principal fibers, and in some area we also see that this PDL principal fibers and they have the close interaction with this the cementum or this side is a zero bone. So it shows the promising that this like strategy may work to like in the center forms a well-organized principal fiber, but on this two ends they'll form the sharpest fiber. Okay, so I'll give a summary. So for this study, I will first did a quantitative analysis of the sharpest fiber, use the right models, and then based on these parameters, we developed a biomimetic matrix to induce the PDL principal fibers and sharpest fibers formation. And the preliminary data, especially in vivo study, shows the PDL principal fibers regeneration and also the sharpest fibers insertion. So it seems like this bioengineering strategy works. And right now we're still working on it. So I'd like to thank my lab members and also my collaborators and finally the funding support. So with that, stop here. Thank you.
I think we can have time for just one question as we switch presentations, please.
Yes.
So in the first model of the vertical, it's right to the physical air, then it's a bio for present. What is it holding the final position?
This scales is made of gelatin. So the entire like microscale is gelatin, and the gelatin is costly. As we implant in vivo, it will slowly degrade it. So here I just showed the result at 4 weeks, and by changing the cross-linked density, we can make the gelatin degraded with a few weeks.
All right, thank you very much. So our next presenter is Dr. Zoe Zhu from Tufts University. Thank you, please.
So good afternoon, everyone. My name is Zoe Zhu. I'm actually a postdoc research fellow from Tufts University School of Dental Medicine. It's my great honor to give you the presentation here. So today my topic is specific long non-coding RNA promotes bone regeneration and eliminates periodontitis. So we have no conflict of interest on this project. As we all know, long non-coding RNAs, short as link RNAs, regulate gene expression at transcriptional, RNA processing, translational, and post-translational levels through interaction with DNAs, RNAs, and proteins. The link RNAs act as essential epigenetic regulators for many pathologic process. And the link RNA anion is antisense long non-coding RNA in human. It has been proven to be a genetic risk factor for atherosclerosis, periodontitis, diabetes, and cancer. Here we can see decreased anion level was found in periodontal ligament cells of periodontitis patients. And the downregulation of the anion inhibits the osteogenesis differentiation of periodontal ligament cells. And another study showed anion is also downregulated in the peripheral blood of periodontitis patients. In addition, in the picture in the middle shows annual associates with periodontitis in different European populations. So taking all these findings together, we come up with the question, what is the role of annual in bone regeneration and periodontitis? However, most of the related studies are based on the clinical study or rely on the periodontal ligament cells from human patients. So to go further on the mechanism investigation and gain a better understanding of the regulatory role of annual, the animal models, for example, gene knockout mice, are needed. So we utilize the annual's counterpart in the mouse genome, which numbered as AK148321, and we named it lincRNA APDC for its major function in atherosclerosis, which is A, and periodontitis P, diabetes D, and cancer C. So basically, the unreal in human equal to the APDC in mice. Okay, so first we detected the expression level of APDC during bone regeneration in mouse bone marrow stem cells. We found the APDC was upregulated during the osteogenesis and was downregulated during osteoclastogenesis and adipogenesis. And we also observed the increase of aerial expression during the osteogenesis in human fetal osteoblast cells, which is consistent with the mouse results. So these results indicate lincRNA APDC is involved during the bone regeneration. Then we established the APDC knockout mouse and APDC overexpressed cells for the further investigation. The success of the gene editing was verified, shown as here. We harvested the bone marrow stem cell and the osteoblast cell from the cavalry bone from the APDC knockout mice and the wild-type mice, and cultured them with different inductions, respectively. We found the osteoblastic biomarkers and the mineralization level of the KO group. The knockout ones were decreased comparing to the wild type, while the osteoclastic and adipogenetic markers are increased. Here are the real-time PCR and cell staining results. And on the contrary, we also detected in the APDC overexpressed stem cells. We observed the increased osteoblast differentiation and mineralization, and a decreased osteoclastogenesis and adipogenesis. We also performed the next generation RNA sequencing on the untreated and osteogenesis-induced bone marrow stem cells from both the APDC knockout and wild-type mice, and analyzed the data by comparing between different groups. Here are part of the results from the APDC knockout and wild-type mice bone marrow stem cells after 3 days of the osteoblastic induction. You can see the heat map and volcano shows the gene expression difference, and the geo-enrichment shows a very significant downregulation of many bone formation-related gene sites. Here we want to compare and show you the two geo-enrichment results. The one on the left is the geo-enrichment results comparing between the untreated knockout and wild-type group. We can see even without the osteoblast induction, the bone formation-related gene sites are downregulated in the APDC knockout group. And on the right, after the culturing of the in the osteoblast differentiation media for 3 days, the existed downregulation of the bone formation-related gene sites are enlarged, and much more differential expressed gene or are detected in those gene sites. We can see the different N number here shows the like more genes are detected for the differential expression. And the gene set of bone mineralization is also remarkably downregulated on day 3 on the right, which was not showed in the untreated group. And on the tissue level, the RNA extraction from femur and tibia showed decreased osteogenetic markers. And we also found increased rank ligand and increased PPAR gamma APO1 and APO2 appeared in the bones from the APDC knockout mice, which indicates the negatively regulation of bone mice. And here is the immunohistochemistry showed lower OCN level in the bone of 4 weeks old APDC knockout mice. Because we always, we are very interested in the crosstalk between bone and the adipose tissues. So we also collected the adipose tissue samples. And in the white adipose tissue, the adipogenetic markers are upregulated in the APDC knockout group. And the upregulation of the UCP1 showed here indicate the enhanced energy expenditure of adipocyte precursors. So next, we also performed the Western blot with the proteins. So the deletion of link RNA APDC induced the activation of TLR4 and MYD88 signaling pathway during the osteogenesis. And the proliferation of P38 appeared earlier during the osteoclastic differentiation in the knockout group, which indicated the activation of MAPK signaling pathway through the P38. Besides the classic pathways, we also predict microRNA 99A might be a potential binding target of the link RNA APDC. So we then proved it with the direct binding of microRNA 99A and APDC by luciferase assay. After, you can see here on the left, after the co-transfection, we can see microRNA 99A mimics can significantly suppress the luciferase activity, but with the mutant link RNA APDC, it can effectively reverse this repression. And the figure on the right shows the binding site of the microRNA 99A and APDC. And in our previously study, we already discovered that the KDM68 is a target of the microRNA 99A in osteogenesis. So we further detected the microRNA 99A and the KDM6B level in our knockout and overexpressed models. So we found the upregulation of microRNA 99A and the downregulation of KDM6B in the APDC knockout groups. And in contrary, the APDC overexpressed group showed the downregulated of microRNA 99A and upregulated KDM6B. So in summary of the role of APDC in bone and adipose tissue metabolism, we found the link RNA APDC is upregulated during the osteogenesis. And the deletion of the APDC can impair the osteogenesis, will promote the adipogenesis and osteoclastogenesis. And the overexpression of APDC can promote the osteogenesis, will inhibit the adipogenesis and the osteoclastogenesis. The APDC deficiency impaired bone formation, enhance energy expenditure of the adipocytes. And the APDC can affect the osteogenesis, osteogenesis through the MAPK P38 and the R4 MYD88, and also another novel pathway, the microRNA 99A KDM6B. Next, we want to talk more about the APDC in the periodontitis. So as I mentioned before, the lincRNA enrol is a genetic risk factor for periodontitis. So we also found in many reports that the downregulation of enrol showed in periodontitis patients. So we want to transfer this phenomenon into the animal model. So we performed a novel method of the experimental periodontitis model for the periodontitis study in mouse. The original design and blueprint and the 3D file were optioned from Dr. Zhao's lab at UNC Chapel Hill, and we 3D printed this set of tools for our EP surgeries. These tools can help avoid mass bleeding and trauma comparing to the traditional method and significantly shorten the operation time. And you can see on the very right, the lower picture, we performed it on both the right and the left side, which gave us the possibility to collect the samples for both the RNA and protein extraction and also the histology. So we performed the EP surgery on both APDC knockout and wild-type mice. And for the time points, we have 3 weeks and 6 weeks group. And the lectures were checked every 3 days. Here shows the microRNA reconstruction. We can see the bone resorption is more severe in the KO group compared to the wild type group on both 3 weeks and 6 weeks. And in the KO groups, we also observe significant increase of bone loss in female than male. And the histology analysis showed more bone resorption activities on alveolar bone in the knockout group. And we will perform the spatial transcriptional analysis with GeoMax on this tissue slice to further determine the changes in the microenvironment during the periodontitis development. And we also performed the RNA sequencing with the gingival tissue from control and periodontitis groups of both KO and wild-type mice. It's very exciting that some distinct gene expression signature were already identified with the periodontitis affected gingival tissue, but the results of this part will be available soon. So to summarize, the lincRNA APDC promotes bone regeneration through microRNA 99A, KDM6B, and MAPK pathways. And APDC deficiency aggravates bone loss in experimental periodontitis model. And lincRNA APDC has therapeutic potential in periodontal disease. And at the end, I want to thank all my coworkers, Dr. Liu, Dr. Tu, and Alyssa, and of course, my PI, Professor Jack Chen, and special thanks to Professor Thomas Wandak from Fossas Institute for his help on the RNA sequencing and bioinformatics. And thank you, everyone.
So it's also open for a question or two from the audience for Dr. Zhu.
Okay, so our final speaker is our symposium chair, Dr. Marco Bartolini, and he's from the University of Michigan, and he'll talk about the personalized and the tissue-specific melt inactual right-hand scaffolds for coordinated regeneration of soft and hard periodontal tissues. So, Mark.
Kahua. There is a So thank you so much. So it's a great pleasure to be here among friends, and I thank again all the scientific groups for co-sponsoring this symposium, and also the scientific committee for giving us the opportunity to share our overall work in both biological aspects as well as materials innovation in regenerative periodontics. Let's see. Oops. Nope. Okay. Okay. All right. So this is really a snapshot of the activities in my lab, particularly within the context of innovations within craniomaxillofacial bone, as well as periodontal tissue regeneration. So over the years, over the past 12, 15 years, we have been working quite a lot and extensively using different type of scaffolds, primarily electrospun-based scaffolds, where Dr. Liu already showed the opportunities within electrospinning technology. So we have been expanding our work within the area of periodontal tissue regeneration. And now, more recently, at the University of Michigan, we were able to acquire a pretty heavy, highly translatable 3D printing, bioprinting platform, where we can really take advantage of the convergence between technologies, meaning the ability to print a ceramic material that can potentially mimic the bone architecture, the bone structure, the bone composition. So we are looking into that right now. At the same time as we can work with soft materials such as polymers and polymeric based materials to envision and devise soft tissue mimics, for example, the periodontal ligament, where we can tune really the polymeric composition to achieve the degradation that that is needed to be able to replace or regenerate PDL in one side of the interface, and perhaps on the other side, foster the regeneration of bone. So this is just a snapshot where we really take advantage of all these technologies within the biofabrication arena in periodontics, oromaxillofacial surgery, orthopedics, and also then the generation of these defect-specific personalized scaffolds for both craniomaxillofacial complex regeneration, bone regeneration, as well as periodontal regeneration. So it's as a little bit of a background, the last speaking in the symposium, I think we talked a little bit about periodontitis and the consequences. So it's really safe to say that one of the main reasons why we need to advance our understanding on the field of biomaterials is because of the aging global population. We have an increased number that will result from a lot of people suffering from missing one or more teeth. So there will be quite a lot of a need for surgical and restorative procedures. And this has been done primarily in the area of periodontal regeneration using the barrier membranes, guided tissue regeneration, and guided bone regeneration principles, where for many years and to this day, these membranes, resorbable or non-resorbable, are used. They are placed in between soft tissue and protecting the bone defect. So essentially prevents epithelial tissue downgrowth and fibroblast transgrowth into the defect, while maintaining the space and allowing progenitor cells to essentially give rise to the periodontal tissues that were lost due to the disease progression. So the way that we have envisioned over the years has been giving another look into the ability to regenerate periodontal tissues. So not only taking advantage of this well-established technique through GTR and GBR concepts, but more importantly, the use of personalized type of scaffolds. So how can we understand what is missing and then print the missing part? And this missing part essentially has different tissues that compose the periodontium, and how can we then use different type of materials, even different architectures, to be able to simultaneously regenerate bone, cementum, at the same time that we are trying to regenerate periodontal ligament that was already highlighted by Xiahua in his presentation. So as I said, we really started in the field of periodontal tissue engineering using electrospinning, and we really over the years did numerous studies leveraging all the ability of electrospinning to allow us to use to make scaffolds, nanofibers-based scaffolds, with the incorporation of bioactive substances and molecules such as growth factors, inorganic materials such as bioceramics. But the problem of electrospinning is that no matter what you do, the technology leads you to a very low freedom in building three-dimensional structures. So this is very simple to manage using electrospinning, where we spin these fibers into a mandrel. This mandrel usually is modified with a Teflon coating, so we can slide out of this mandrel, this 3D architecture. So we have been using this for larynx and trachea regeneration. So that's something that we've done over the years, but primarily for periodontal tissue engineering, where we are trying to build interfaces with defect-specific architectures, this poses a significant challenge. I already mentioned that the electrospinning allows us to modify these polymeric fibers with the use of bioceramics, for example, with the use of cytokines and growth factors, all to enhance osteogenesis or potentially PDL formation. And one of the early works from our lab really identified the ability of generating these nanofibers with not only the specific chemistry, meaning the composition of these polymers that potentially could house different molecules and antibiotics, for example, or could be modified with bioceramics, but addressing the tissue specificity of these periodontal defects. So here we have just a cartoon showing the schematic of a bone defect that will go through bone regeneration by using a membrane in addition to bone grafts. But our question at that time was, how can we make a tissue-specific, layer-by-layer, multi-layer membrane could be even displaying therapeutic properties? So we did that through sequential electrospinning. So we see layer by layer, we put layer by layer of these nanofibers, forming a membrane such as seen in this cartoon. And this is essentially the ability to make this epithelial tissue area where we were, for example, incorporating an antibiotic such as metronidazole. And on the bone defect, we were making these fibers modified with nano hydroxyapatite particles. So this was published about 10 years ago, but really was the starting point for us to be envisioning the area and the use of biofabrication into periodontal tissue regeneration. So last year, we published a pretty extensive review paper on international materials reviews, where we addressed pretty much everything that is going on in the field or has going on in the field for the past 10, 15 years, and how really the idea and the ability of converging different technologies to form tissue-specific scaffolds aiming to regenerate the periodontal tissues could be done using 3D printing, 3D bioprinting. The idea here in the platform that we have the ability to print, for example, polymeric, very sturdy scaffolds. And this is really mimicking or the ability of making scaffolds such as fused deposition modeling, or also selective laser sintering. So this is, really sturdy, really robust scaffolds that we can, even though tune porosity and architecture, really lacks the ability to print at a very micron level that can help, for example, guide periodontal tissue formation, periodontal ligament formation. So what we do and what have we betted on and we are exploring more closely is a technology called melt electrowriting. So the technology essentially derives from electrospinning, where we have a polymer. This polymer can be dissolved by heating, or it can also be dissolved using a solvent. And in our case is through heating. So the ability to apply an electric voltage at the tip of that needle allows us to form a polymer jet. And this polymer jet then can be used to write different three-dimensional structures. So we, as you can see here, this is the polymeric jet that is forms as you optimize the electric voltage that is applied into this polymeric solution. And then you have this very thin filament that can then be able to work through whatever architecture and provide a layer by layer assembly of these filaments to generate different type of three-dimensional architectures. So initially, so it's been almost 3 years that we have had this technology, and our work has evolved over the years. And it started initially as to be able to print these structures and try to understand what polymers can we use. We started off using polycaprolactone. We are able to make these very well-defined architectures, cells like the polymer. They don't necessarily suffer from the ability to attach and spread onto these fibers. So biocompatible, as we have seen PCL process from using other technologies. So we are able to see that these scaffolds can be biocompatible. And this is what led us to our next step. Two stories also in one presentation here. The first story is how we could take an approach that is similar to what is used in civil engineering, where you take concrete and reinforce with this steel fabric to make a material that is stronger than its individual parts. So this was led by a postdoc in my lab, Dr. Dubé, and was a project where we were envisioning the ability to generate guided bone regeneration membrane based on a hydrogel. How could we reinforce a hydrogel, which essentially lacks quite in the mechanical competence, but using these fiber meshes, we were able to making into a possibility for future clinical translation. So the idea was to use melt electrowriting to make these fibrous meshes with different porosities or strand spacing here. In this case, the strand spacing here is 500 microns. And the idea was to use methacrylated gelatin hydrogel, modified or not, with a bioceramic, in this case, amorphous magnesium phosphate, and then investigate the influence of adding these fibrous meshes in terms of not only mechanical competency of these hydrogel materials, but as well as the ability to function as a barrier in vivo, as we demonstrate here. So we were able to implant in a critical size defect these hydrogel-based membranes reinforced with these melt electrowritten meshes, and see quite a significant bone formation after 8 weeks in vivo. Just I want to point out for a cross-section of this hydrogel-based reinforced membrane, where we can see the the thickness of the hydrogel material. Actually, within this hydrogel membrane had a thickness of 1 mm. So essentially, you have a 500 micron in height material, fibrous mesh, that was able to be embedded into this hydrogel. We understood that the thickness of the hydrogel would play a role and demand more meshes or a mesh of a higher thickness to be able to reinforce the hydrogel material. And we can see here, when we place 4 meshes within the same thickness, we were able to almost a 4-fold increase in the stiffness of this hydrogel. So this was really the first study of our lab that was published using melt electrowriting to make these meshes and potentially be used to reinforce hydrogel-based materials. The second part of the use in our work in melt electrowriting came about with also Nilesh, but also more important with Arwa Dagheri, that was a PhD student in my lab, recently graduated and took a position back home in Saudi Arabia. And she was really the driving force of this second strategy that I'm presenting here, using melt electrowriting to regenerate really tissue-specific bone zone, PDL zone, to make a scaffold that could address and potentially coordinate the regeneration of both tissues simultaneously. So the first idea behind was, how can we take PCL, a material that has a well-established biocompatibility, but does not necessarily really support osteogenic differentiation or bone formation. So how can we take an inert material to bioactive? So the first study in our lab was really devising a coating to coat these melt electrowriting fibers with the fluorinated calcium phosphate to really enhance the ability of osteogenic differentiation. So we were able to use a biomimetic type of immersion of these PCL fibers to generate a fluorinated calcium phosphate coating. So we studied from atomic force microscopy. We were able to understand the influence of the coating in terms of roughness, the influence of the coating in terms of the mechanical properties of the scaffold, and that's what I'll be highlighting in the next slides. But the first work started really to laying the groundwork for these tissue-specific scaffolds that I'll show in a minute. So the initial approach was really to define what is the best strand spacing that will allow increased cell attachment and cell proliferation. So we demonstrated that for this non-coated fibers, so this was before coating these melt electrowriting fibers. So the 500 micron strand spacing was the most ideal, the one that favored, particularly in the initial days, best cell attachment and best cell proliferation. Once we defined this overall architecture, 500 microns strand spacing, then that was the architecture that then was subjected to the coating formation. So we checked, for example, what happened when we promoted the coating formation into these PCL scaffolds, and we show here at tensile testing, the presence of the coating led to enhanced in mechanical properties. We were also interested into understanding the coating stability. So for how long this fluorinated calcium phosphate coating would be potentially favoring osteogenic differentiation of in our case here, periodontal ligament stem cells. So this coating was stable, and we did some TGA experiments that show that was stable over 7 days. And at the same time, I didn't put the data here, was releasing calcium phosphate ions also. In terms of cell proliferation, the coating also demonstrated to be non-toxic and allow a pretty consistent cell proliferation over four weeks. Bioactivity, we demonstrated that the coating was bioactive or is bioactive through SBF type of studies, and we show here the data. Also, the coating was resulted also into osteogenic activity of our PDL stem cells. So we can see improved osteogenic activity by both ALP and gene expression. So the blue bars represent the coated scaffolds, particularly in the absence of osteogenic media. The coating per se was able to guide and drive osteogenic differentiation. Then, as shown before by our colleague, Professor Liu, we also went on and placed these scaffolds into a fenestration defect in rat. And we were able to demonstrate that even though the architecture at first could lead to thinking of what is the role of the porosity in, for example, preventing soft tissue infiltration, epithelial tissue infiltration. So we were able to demonstrate that this scaffold per se, when placed into these defects, were able to lead to the regeneration not only bone, but also PDL as shown here. So the presence of the coating or these scaffolds coated with this fluorinated calcium phosphate led to significant bone formation compared to the non-coated counterparts. So this was the first part of the story, particularly also of her PhD. So trying to take a material that is inert, making into a potential osteogenic material to allow us to then think about tissue specificity, a zonal type of scaffolds using melt-electro-writing as a personalized strategy to build these scaffolds. So this paper is currently under review, the data that I'm showing here. But the idea was then how to combine the bone zone that we optimized in the previous paper, in the previous study that I just went on to share the data, with the ability of printing these fibers parallel to each other and can be oriented in a way that we have freedom to orient it in a way to mimic PDL architecture, and that would potentially support PDL formation. So we study different scaffold architectures, as we can see here, to understand the influence of these aligned fibers in PDL formation. So we process a random fiber just to as a control group to hypothesize in terms of the alignment, the role of alignment in PDL zone, PDL formation, but also we explore other architectures to understand what could that lead to. So initially, again, just a very simple experiments in terms of cell proliferation, trying to understand the different architectures favoring attachment and proliferation. So interesting, showing the orientation of PDL stem cells over the long axis of the melt-electro-written fibers. So that was one important finding. And then due to the lack of time, I'll just wrap up with 2 more slides showing our initial findings in terms of making these scaffolds using the same protocol, using the same equipment, that we could at the same time print a zone that is bone-driven zone or bone supporting zone attached to the other side, the other interface, to foster, encourage physiological regeneration of periodontal ligament. So we also again use the fenestration defect model, and the group tests included also a group where we so these are biphasic, so these are 2-layered 2-zone scaffolds. So the bone zone is the same configuration, the same composition as in the previous work. On the other side, we have then these aligned fibers helping a PDL formation. One hypothesis that we had, or one idea that we had, was to try and prevent potential soft tissue infiltration. So we added gelatin hydrogel to try and promote, to hinder soft tissue formation. So, and then we implanted these different scaffold types. So there was a sham group, a collagen only group, And then the aligned fibers on one side with the fluorinated calcium phosphate on the other side, and the aligned fibers on the one side, the fluorinated calcium phosphate on the other side, with the addition of the whole scaffold submerged into a collagen hydrogel. Interesting enough, we were able to determine that the collagen containing aligned fluorinated calcium phosphate scaffolds supported increased bone formation, as we can see here in this graph. That was not necessarily true in terms of supporting PDL formation. So we understood, based on the histological data, that perhaps the collagen covered and made an envelope into this parallel oriented fibers, impeding cell alignment and physiological regeneration of PDL. But all in all, we were able to demonstrate then that a scaffold with aligned fibers with fluorinated calcium phosphate were able to generate periodontal ligament fibers connected to bone, particularly over the top here. So in summary, and this is a next steps of our project in terms of really generating these personalized scaffolds using melt-electro-writing the way that we have here, the defect micro-CT of the defect, the generation of the defect using CAD software, and then being able to then generate the prints that were used here using melt-electro-writing. So in summary, we were able to make polymeric tissue-specific scaffolds via melt-electro-writing. These tissue-specific scaffolds were able to guide in vivo soft and hard periodontal tissue regeneration. And we also measured the accuracy of the prints. So the accuracy of the prints in relation to the architecture of the defect was about 70%. So with that, I would like to thank my lab and my collaborators and the funding. And I think I went over my time, so I apologize for that. But I think the four of us will be here to answer any questions. Thank you all for coming, and we are ready. We'll be happy to answer any questions. Thank you.
I noticed that when you make the scaffold, the PCL and the borrow contact, two stages scaffold, right? Also, you make the PCL and podium. Is it possible that you combine them together, use one step, still use the wet electrolyte?
Yeah. Great question. So we have just a paper that just got finished, and my postdoc is in the back, Renan. So we have a paper coming out where we bypass the coating, right? So the coating would prevent us from taking the other side, the PDL printed side. How can we isolate the other side from a coating process? So right now we are able to generate PCL fibers doped or impregnated with magnesium phosphate, for example. So now we can use just two different polymeric solutions. One that is a paste PCL magnesium phosphate. The other one is just PCL, for example. We've been looking now into other polymers, actually, not only PCL, because of the degradation profile, the slow degradation profile of PCL. So I've been collaborating with Matt Backer at Duke University, and he has pretty interesting polymeric materials such as polyester ureas modified with amino acids. So those are particularly of unique opportunity for soft tissue regeneration. So we are now expanding the use of other polymers, or polymers that we can really tune for the timing of the regeneration that we are trying to achieve. So trying to understand the necessity of having perhaps different type of polymers I think it's important also moving forward. So thank you. Thank you. Sure. Sure. No, this is a great question. So we were able to demonstrate that we can, for example, print these fibers at different positions or at different configurations. So parallel fibers, we can orient in whatever angle. So we are studying now angulation effect into PDL formation, for example. We were able to demonstrate that we can, through a coding process, for example, change texture. So there are opportunities that we can enhance, for example, attachment, right, of newly formed PDL tissue, for example. So I think there are more for us to unveil, but the technology, I think, it has not only the ability to use different materials and formulations, but I think the other layer is really the ability of using imaging, right, of the defect to be able to generate really the replicas of what is missing and being able to implant it. Hi.
Hi, Bottino. Hi. I'll ask people to use the microphone because we are sitting in the back. We cannot listen the questions properly.
Sure.
But anyway, this was a fascinating symposium. I'm Cristiane França from OHSU, and I have two questions, one for you and one for Dr. Liu. Bottino, what is the scalability of your fascinating process to engineer tailored tissues for regeneration?
Sure. No, thank you, Cristiane. So it's really highly scalable based on what we know so far. So, for example, when we did this initial studies, the paper that I alluded to with the coding, so we were able to print as many as 24 type of scaffolds in one batch. So imagine for in vivo animal studies, the reproducibility of the same scaffold. So we are able to make, for example, 24 of them using the same polymer in one shot. So scalable, so it takes a few hours to print all of them. In terms of the architecture, is really reproducible. So we can essentially, it's a printing mode, so we can do that. So what we are doing right now, it's really taking off of, so the field of melt-electro-writing, as you know, and now Paul Dalton is in Eugene, near you guys, and he's one of the inventors of this technology. So if you look at the literature of melt-electro-writing, 99% of the papers are with PCL. So we are now endeavoring into a different area of other polymers that can be more suitable for what we do in dental medicine, regenerative dental medicine. So I think there is the scalability, there is the ability to make defect specific, tissue specific, and I think it's a combination of also technologies, the way that I alluded in the beginning, where we are making, for example, bone mimics, because we are developing our own bone pastes in the lab to print different type of bone, ceramics. So I think there is quite a lot there. Xiaohua.
You have the same question? Oh, okay, I saw the same question. Okay.
Thank you, everyone. Thank you.